Welcome to Research Renaissance, presented by the Karen Toffler Charitable Trust. Embark on a journey into the forefront of cutting-edge brain science as we feature insightful discussions with a diverse array of guests, from early career researchers pioneering groundbreaking science to influential voices in investment communities, policymaking, and research institutions. Together, we delve into neurological diseases, their causes, treatments, and the quest for cures. Join us in advancing our understanding of the brain and addressing its ailments. Welcome to Research Renaissance. I'm Deborah Westphal, your host and guide through the fascinating future of the brain. Joining me today is Dr. Ignaty Leshchiner, Associate Professor of Medicine at Boston University School of Medicine, where he specializes in computational biology and cancer bioinformatics. He is also a 2024 Toffler Scholar. Today's discussion is focused on the relationship between cancer research and the research of the brain and its diseases. So let's dive in. So welcome.
Hello. Thank you for having me.
So excited to have this discussion with you. It's you're working across these very different areas, namely cancer research and then Alzheimer's and dementia research. And this is kind of a new and really new and exciting research area that few have embarked on now. And so I would like to just kind of just start with describing what is this relationship between what you do on the cancer side and the bioinformatics side and the research of the brain.
Yeah, I think it's nice to start with the connection of some of the research projects that we did early on with studying brain tumors, specifically glioblastoma, which is one of the most deadliest tumors, unfortunately. And the way I got into it, that I had some also family members that were impacted by this cancer. And the fact that it's one of the least treatable cancers and 95% death rate within a few years is really an important cancer to study. And in fact, there's, I think, over 120 different tumors of the brain that exist, and not just glioblastoma. And some of them are more benign, some of them are more aggressive, like GBM, glioblastoma. But it's remarkable that brain is one of the most important organs, I think, to study in terms of cancer research. It is hard to study brain tumors, mostly because it's really hard to biopsy or get a sample tissue from the brain when the person is alive. So my lab is really focusing on designing methodologies to sample tumors in a non-invasive way, where you can study the genetic makeup and the evolution of the tumor without having necessarily to do a recurrent biopsy and sample the tumor physically after surgery. We have studied glioblastoma, which is very resistant to treatment, and we found that unfortunately it progresses relatively quickly, but it also evolves as it progresses. It's one of the most quicker evolving tumors, and it spreads throughout the brain. We did find that in our understanding is that the cells that evolve actually spread throughout the brain relatively early. And then by the time people see recurrence, it's actually pre-existing small kind of focal lesions that actually have already spread through the brain. So it's very complicated and important topic. There's very little drugs at work. There's some chemotherapy that is used for initial treatment, temozolomide specifically, and then there's some radiation treatment that people receive. It's still not very clear what is the predisposition of glioblastoma. It is a relatively early onset tumor, so a lot of patients get sick within their 40s and 50s, which is not typical for most other cancer types. And it is believed that there is some strong familial component, so basically, genetic predisposition to GBMs. So I think that's the beginning of the connection, and the methods we're developing for probing tumors and probing other tissues within the body in a non-invasive way, mostly by looking at circulating nucleic acids in the different fluids, specifically blood. And if we're talking about brain diseases, CSF, cerebrospinal fluid, that collects a lot of the material throughout the brain. Blood is one of the most non-invasive ways of actually studying the genetic makeup of the tumors on different organs, and we're really interested in using that as a biomarker of both early detection of disease states, but also monitoring progression and response to treatment, which are both very important in cancer, but also in Alzheimer's disease and other neurological disorders.
So you said, this non-invasive way, and this is possibly one of them is through the blood. This is a fairly new area of research. Is that correct? Is by using blood as maybe a biomarker or possibly a diagnostic test for what is going on, and whether that be cancer or whether that be the aging process or diseases of the brain, other diseases of the brain. Is this a fairly new area of research?
Correct. It's a relatively new area, even for cancer monitoring, probably less than 10 years, 7 years in the making in terms of the first big studies that looked kind of holistically across different cancer types and diseases. There's much more interest in aging and clonal evolution of tissues in the recent years. Literally a few years ago, this whole process has started. Specifically looking at circulating DNA and RNA, people have been and are looking also at circulating proteins. And there have been some very interesting recent reports, specifically on Alzheimer's disease, and actually monitoring disease states based on circulating peptides and circulating proteins in the blood and CSF. We believe that you can achieve a higher resolution, higher sensitivity about disease states from actually circulating DNA and RNA. They carry both genetic information, such as mutations, but more importantly, they carry epigenetic information, methylation, which are modifications on top of the nucleotides that control gene function. And they're known to also be modified within age in different organs. That, first of all, allows us to identify pieces of DNA and RNA that come from specific tissues, because that signal is tissue specific, and also see the state of the DNA and RNA in those tissues within throughout the change in age and disease state and response to treatment. In terms of tumors, we also see the genetic evolution of those tumors throughout the life history of the tumor.
But we don't have these blood tests right now. Is that we couldn't, the general public couldn't take a blood test to try to understand? Is this something that, am I understanding that correctly? Is this something that has to be developed because we're just starting to understand that? Or are these tests out there for people to take the test to try to understand if they have a progression of a tumor or some sort of neurological disease?
Correct. So most of the tests are not available. They're still in development or in R&D stage. Some are entering clinical trials right now. There are some early tests that are available from the blood to monitor evolution and response to treatment. So companies that are marketing those, they're mostly looking at mutations that occur or have been in the tumor. There are new areas of those tests that are entering the clinical space right now, which we call minimal residual disease tests, that are actually trying to not just detect DNA in general in the blood, but actually measure it to very, very low levels when, for example, the tumor had been removed through surgery and there's no real large amount of genetic material left in the person. But still, if the tumor DNA is detected in the bloodstream, it means that the tumor is not eradicated yet, unfortunately, and that means that it's most likely to recur. And there are several clinical trials that have been showing that there's a benefit of knowing if there is any residual tumor left for choice of treatment, and also for monitoring when the patients need to basically switch treatments or need to look for other options. There are some FDA-approved companion diagnostics also that are starting to enter the clinical space. For example, right now, there is a recent trial showed in colorectal cancer it's beneficial to do these MRT circulating DNA tests to detect people that are positive for leftover tissue, and those would receive chemotherapy, versus people that are negative. Actually, it's recommended not to receive chemotherapy if they don't have any other complications, because it doesn't show to be beneficial in any way to actually receive additional therapy at that point if the MRD test is negative.
So you started with kind of the personal, connection to somebody in your family that had this, these brain tumors that are very hard to detect, and that they're very progressive, and, and it's not likely to, to survive this. Tell me a little bit kind of your personal journey with this. Was obviously kind of a moment of influence, but what was that journey, and why did you choose this area of research? And then, what was it in that research that kind of led you to a broader kind of perspective of the brain, and the connections with blood and other kind of body tissues?
Yeah, unfortunately, through the years, there were several close friends and family members that had different types of cancer, and several of them had brain tumors. And different cancers give typically different scenarios of the patient treatment plan. And some cases are somewhat more benign. In others, people have 5-year outlook of survival or more. In case of glioblastoma, it's unfortunately typically less than 2 years. Very quickly moving tumors, and even when you try to do some genetic analysis and try to understand the specifics of that tumor in a specific patient, it's very hard to choose treatments. We don't really know right now what are the general guidelines of, if you find a genetic mutation or you find some sort of amplification of some gene, what drugs. We could just suggest which drugs might work. There's no rigid guidelines in terms of trying any new therapies or clinical trials. Even though when people typically run out of treatment options, they often try to look for new clinical trials or new therapies that are being investigated currently. Immunotherapies. There's some recent exciting reports about CAR T therapies in brain tumors. So all of these things are required typically, knowing the genetics of the tumor, which could be obtained through either surgery or in our case, through blood biopsy or CSF biopsy, which could facilitate enrollment of patients in these trials. For example, if patients have EGFR amplifications, which is a known cancer driver, there are some these new clinical trials that are exporting CAR T therapies or other targeted therapies against EGFR. Maybe to kind of give a bit of a perspective, when some of these interesting research started, there was an interesting small successful study that came out from the lab that I worked in at the Broad Institute, which studied a non-malignant, a benign tumor. It's a rare papillary type of tumor in the brain, craniopharyngioma, which is not typically aggressive and spreads, but it has this complication that it grows relatively uncontrollably. And, it can be removed through surgery, but then it comes back and continues to grow. It doesn't spread to other parts of the body like other more aggressive tumors, but it still carries a lot of issues with, because it's actually inside the brain. So people have all different neurological symptoms, and they can lose vision. They could develop all other aspects from the tumor actually pressing on the brain and damaging the tissue around it. In that study, there was sequencing done on that tumor in a patient, and it showed that the patient had a BRAF mutation, which is another oncogene, another cancer gene. And we know that there is actually a drug against a BRAF. It's called a V600E, specific amino acid change modification in an oncogenic BRAF. There's a targeted therapy that is used in melanoma, BRAF positive melanoma, that was actually applied in that case. And the patient fully recovered since it was not a malignant tumor, which has the ability to evade treatment and evolve very actively. Benign tumors in these cases are often more treatable. They're actually not able to develop resistance that easily. And it turned out in that patient, which had multiple surgery and was losing his vision and had multiple other complications, actually was basically a solution for him. The tumor completely went away and the person completely recovered. So it was kind of very remarkable and a direct connection to the science that we're doing. Unfortunately, it's not always the case. There's usually many years passes between the science we're doing right now and available therapies. But in this case, it was a early kind of sign of where genetics are actually contributing directly to patient care. That was more than 10 years ago, but it was I think one of those remarkable studies that really got my interest into studying both brain tumors and now more other neurological diseases. I think it's very important since the brain is a unique organ in many aspects in terms of the blood-brain barrier and therapies. A lot of the therapies that are developed for other tumors or other conditions are not always usable in the brain. For example, there are a lot of antibody drug conjugate therapies that are now being developed for different tumors. They're much harder to use in the brain. There's, less penetrance into the brain, and there's specific formulations that need to be developed for these drugs to be available in the brain. And in a situation when maybe this tumor would have been outside the brain, it would be much more treatable. But that's not necessarily the case in brain tumors. And that's also true in Alzheimer's disease treatment, because some of the new promising therapies that are, anti-amyloid antibodies have been recently approved, and some others are being developed that are showing first clinical significant progress in actually treating early Alzheimer's disease. Also, obviously, they need to be developed as accessible to the brain, and so it's a very important aspect. We're hoping with our diagnostic approaches that help to monitor what's happening with the brain as the treatment is going on, to be able to see the effects, the actual changes, biological changes and expression changes in the brain that happen as a successful treatment is occurring, so we could infer what a successful treatment would look on the molecular level without obviously getting any physical tissue from the patient, which is obviously almost always not possible, really. So this really non-invasive way of actually knowing what's happening with the cells is very exciting. These methods are new and novel. We're hoping that we'll be able to uncover a lot of biology behind it.
So am I understanding this correctly then? If we can understand this brain barrier, which it's, the blood flows through the brain organ to other parts of the body, and we can test that. We can test it in a non-invasive way. That, kind of the first step that you talked about is looking at how a treatment may be working or not working. In the future, would we be able to go annually and check our blood and see if we might even be, progressing to a disease or progressing to a condition or to growing a tumor? Is that something, am I understanding that, it's a long time away, but is that something that is possible?
Yeah, we're actually working as a community towards those goals, and I think they're actually achievable. So the first step, as I described, is really to monitor patients that already have cancer. So we know they have a tumor, and we can monitor what's happening with that tumor in a very sensitive and specific way, actually, through the blood. The next step is really to broaden those tests to detect tumors early, which we call multi-cancer early detection tests, which have not been yet really approaching the clinic. But there's several companies and then there's a lot of academic research in this space, with the hope that we will be able to soon tell patients if they do have a tumor and in which location they have a tumor, so they can have follow-up tests and identify the location of the tumor, how aggressive it is, how it's likely to progress. It is a complicated field of study because everybody through aging, they actually have multiple clonal expansions, or basically, you wouldn't call those tumors, but basically sort of dysplasias or growths of clones in different tissues across the body, which somewhat mimic the behavior of early stage tumors. So really distinguishing between early stage tumors that are malignant and are likely to progress to aggressive cancers versus benign growths that are really not dangerous and really don't need a follow-up. That's where the science is right now, is basically being able to distinguish between those two conditions. For example, maybe a simplified example would be, people have moles on their skin, which are often also clonal growths, and they do actually have mutations, and in some cases they actually have oncogenic mutations in them, but they're not dangerous. They grow to some size and they stop growing and they just hang out there, and very few of them actually progress to anything dangerous. So distinguishing of similar processes inside the body, which occur to a degree, is important. So the test is actually sensitive to cancer, because when you start screening millions of patients, you will come up with a lot of false positives if you're not having a very sensitive test to the condition. So we need to be sure that we're not nominating millions of patients for additional screening when they actually don't have cancer. So I'm actually pretty optimistic that within 5 to 10 years we'll have those tests available, and people would actually have yearly blood work done, with a high chance of catching cancer early. And in some way, I would say this is probably the main breakthrough that we can achieve in the next 5 to 10 years with cancer epidemiology, basically by detecting cancer at the T1 stage, the early stage, when it's still treatable and still resectable. We can maybe half the current death rate from cancer, which would be fantastic if we could do with treatments. But the history of the last 20 years doesn't really tell us that we can easily find treatments for all different cancer types. Versus this early diagnostic can really move the needle in terms of actually preventing a lot of the cancers going to the later stages and actually saving people. So we're very, very excited about these types of technologies.
And that's across, when you mentioned the cancer, this is across different kind of cancers across the body. Is that skin cancer, other organs? Is this kind of all inclusive, or is it really focused on certain areas of the body?
Well, that's the main goal of these tests, is that they're multi-cancer indication, that it will almost capture, maybe if not all cancers, a majority of the cancers, because it's almost impossible to require patients to do 15 different tests or 50 different tests. There's over 250 different cancer types. It's very unlikely that people will do more than one test a year, maybe a couple of tests. But the goal of the test is to really catch all possible tumors, and obviously we'll have different sensitivities for different tumor types at first. But the goal is to develop that further and really at least address the most deadly and the most frequent cancer types first that are prevalent, and that would especially some of those that are very poorly treatable at later stages, would really, could really decrease the death rate from those cancers, because some of the cancers are quite treatable in the earlier stages and almost not treatable at the later stages. So the goal is really to have a universal test for all patients, for all ages, and to be able to detect cancer when it's still early stage.
So as you study kind of this area of the blood and how it can detect different things that are happening in our bodies, namely like cancer, the crossover of understanding is also then to other maybe like neurological diseases, because you've talked about Alzheimer's or dementia. And, as we get, the way I'm understanding what you're saying is the more we understand about the relationship with blood and that circulatory through our bodies and what's happening there and how we can detect different things that are going on in organs or in our tissue, that possibly that then gives us a more understanding for neurological diseases. Is that correct?
Correct. And I actually think it probably expands to almost all common diseases that we're studying. Most importantly, I think these blood tests, and we're talking about basically sampling genetic material, both DNA, which encodes the genome, and RNA that codes specific genes that are then made into proteins from the blood that collects this material from either dying cells, and cells die throughout the life of the person. Some die very, very frequently and cycle, and others, like in cancer, they just continuously grow and die all the time, but also in normal organs. And when they do, they release that genetic material in the bloodstream. And also, there's other processes like exosomal release that release some of the genetic material in the circulation that allows us sort of in a non-invasive and very removed way to actually study the tissues of interest without having to take a physical slice of that organ or the tissue, or a needle, poking a needle in some area in the body. And it's very important, I think, both for biology, not just for clinical development, but for the biology of the disease and biology of natural aging and biology of human health, because this is the first time probably in many decades that we can actually start understanding what's happening in the human population, not using animal models or other laboratory techniques, but actually directly measuring patients with disease conditions, healthy individuals, and getting information about what's happening with age, what's happening as disease progressing, what's happening under treatments, and really infer the state of these tissues if we were to take a physical biopsy or physical sample. And that's how we also train these algorithms and understanding, is by comparing what's in circulating DNA or RNA versus the actual tissue. And the project that I have on Alzheimer's disease in the lab is actually comparing the actual tissue of patients with Alzheimer's disease, their CSF fluid and blood, plasma, nucleic acid content, and trying to understand what we can see in common between these media, and how sensitive is the blood to detect specific conditions, how to specific genes. People are starting to infer expression, basically the biological state of individual cells, from just sampling the blood and trying to understand what's happening with those conditions. Plus, we know that the methylation and expression patterns change over age. As people age, they change. And we know that Alzheimer's disease is, in many situations, is an age-related condition. And it's still not very clear when, the processes that generate the plaques, and when they become active. Are they starting from the earlier age, or are there at some point that the astrocytes, for example, that are responsible for clearance of the processed Alzheimer APP or precursor protein parts, but potentially they're somehow modified through aging and dysregulation of their structure and evolution of these cell populations into the wrong direction, if you wish, that break the processes that are normally active in young individuals and cause this kind of condition to develop as people are aging.
So this might give us even insight into the whole aging process and how to possibly counter it, at least to live healthier lives throughout our lifetime, whatever that might be. Is that correct?
Correct. I have a similar project in my lab that is trying to study exceptional longevity individuals, what we call centenarians, basically individuals that reach an age of 100 or more years old. And we know that in many cases, these exceptional longevity is actually running in families. So the siblings of those individuals would also live to a very old age, and their parents. We also know that some of these exceptional longevity patients, they actually have, we can call it, differential aging. Basically, some of their organs age slower than in the normal control population. And there are individuals that have a better function of their lungs, better function of their kidneys, livers, brains, and these traits run in families. And there probably are predisposed by their genome and their genetic inheritance, but also environmental conditions. And we are trying to understand, what does it mean, slower aging for a specific organ, because it doesn't mean that all of these individuals, some of them do have systematic kind of slower aging, but that's not necessarily always the case. And again, by doing this non-invasive sampling, we're able to study even healthy individuals. You will not collect tissue from different organs from healthy individuals, definitely. But by using blood, we can actually interrogate how these individuals are living longer, and maybe we can find, anti-aging medication and understanding the biology of proper aging or slowing the aging process and slowing disease progression.
So can you talk a little bit about your laboratory? I think there may be listeners out there that may not really understand or have never really even considered what a research laboratory is. What type of people do you have working there? Kind of what is the day in the life, if you would, of someone working in your lab, and what are they doing?
Yeah, so I think the academic laboratory environment is maybe very distinct from what are people used to in companies and in different other work environments. It's sort of like this nuclear family, if you wish, of people working on a specific goal and a specific topic. Typically, the laboratory has a PI, a primary investigator, a professor, or that is heading it. But then there's multiple layers of different individuals that are often focused maybe on their different personal goals, but also on different research goals. So we have in the lab, we have postdoctoral scholars that already have a PhD, so they had experience of several years already studying and researching specific topics, sometimes in our field, sometimes in a similar field, and they're switching fields. But the goal of them coming to the laboratory is really developing their independent careers for the future and maybe becoming professors themselves over time. So they're kind of adjusting the training that they received during their PhD to become more of an independent researcher. We have graduate students, PhD students that are obviously pursuing their thesis and their degree. It's typically a 5-year process in the United States. So some are just entering that 5-year period, and they're really doing the courses and getting used to the research environment. And others are more senior people. They're really looking to complete their projects. And some move into industry, some move into postdoctoral position or academic positions. We have a lot of master students and undergraduate students that are really interested in getting some sort of a taste of research and exploring new scientific areas and getting additional skills and just exposure to scientific environment. So my lab is somewhat atypical. We call it a hybrid lab, meaning that I both have wet lab, basic experimental section where we do experiments, but since I'm also a computational biologist, we have a dry lab, basic a computational lab, where all the analysis and modeling and software development is happening. And some individuals within the lab have both skills. They can do experiments and do computational work. Some people are mostly doing computational work. They don't really appear in the lab, in the wet lab. Others are more experimentalists, and they're doing more of assay development and actually running samples and analyzing samples, but they typically try to do also some bioinformatic work. So we call that a kind of a hybrid approach in this sense. Sometimes a laboratory is just working on computational methods or just doing experimental methods.
So there's a lot of talk, a lot of chatter in the news around AI and machine learning and advanced algorithms, and there's hope for these things, but there's also fear. Since you have that computational side, how does today and maybe even kind of a peer into the future, how is that advanced technology, if you would, how does that help you today? Or how might it help you in the future?
We look very optimistic into utilizing the new AI methods, machine learning. We're using machine learning and other computational methods all the time for our tools and trying to understand and infer the biology from our data sets. There's a lot of development in terms of analyzing the bigger data sets that we're not now getting. For example, in the last few years, we started to generate single cell data sets, meaning that now we can sequence each independent cell within a tissue block, being it a normal organ or a tumor. We can now separate different cells and sequence individual immune cells, tumor cells, stromal cells, other soft tissues. And that allows us to really identify the expression pattern, methylation patterns, mutational patterns in individual cell types, both in tumors and normal organs. And this is really a revolution in a way of data generation that happened recently. And that allows to discover new biology that people have not even knew existed, even maybe 5 to 10 years ago. But that also creates an explosion of data, because now we used to have one data point for one sample, and maybe measuring many genes, but still was just one sample. But now for that sample, we can have tens of thousands of cells from one sample, and we can have hundreds of samples. So we're now looking at millions of points of information. And to make any sense of all of this, that's where machine learning methods really help to generalize, extract information, and just being able to make sense of all the data at once. Because a human brain, even with the help of traditional computational methods, sometimes there's just too much data to really grasp the insights from it. So a lot of the AI methods in single cell biology now are focused on really interpretation of the data and identifying patterns and similarities between different cell types, different tissues, response to treatments, predicting if the cell will respond to a specific drug. For example, if the tumor would respond to a specific drug or therapy would work, and how aging affects different tissue types and different organs. And there's many different cell types in every organ. So it's very important because they kind of work as a collective for any of the health and disease issues. So we need to really study the whole picture. And another aspect is kind of a next level that is just entering the field also is actually what we call spatial biology. Is not just studying single cells when they're dissociated and studied separately as individual cells. We're now trying to study single cells in blocks of tissue where we also study the spatial organization and interaction of these different cells between each other. For example, how the immune cells interact with other members of the tissue, and that's very important in both cancer and neurological diseases that are immune mediated. They're important in many other conditions where the interactions of different cells are key to studying the disease condition. And this is really a revolution. And again, it generates terabytes and terabytes of data that we haven't been used to until recently, and to make sense of all that data. And even partitioning a slide into different cell types and figuring out where which cell exists and what expression of that cell type has, you already need the AI machine learning methods to just do that.
And it would seem that visualization of these complex connections is really important. That new kind of technology or capability to do that is going to be even more important.
Correct. It is very important to both be able to visualize and just access all the data because, again, as I mentioned, it becomes very large data sets that need to be effectively and quickly accessed, aggregated. We try to use a lot of cloud computational capabilities, which are, in some way unlimited, or you could store much more data that we used to be able to store in just our lab hardware, and quickly analyze it and train models across that data. And yes, so visualizing and then actually, being able to look at these images with different annotations, making sense of it. Again, it's important because we have different domain scientists. We have clinical scientists that know, pathologists, for example, that know how the tissue needs to look in a healthy individual versus a pathological condition, and they, together with the scientists that study the biology and the genetics, we can make sense of what's wrong and which pathways are dysregulated and what proteins are dysfunctioning and how to maybe treat the condition. All of these tools are built to facilitate both our interpretation and also interpretation of both clinical and biological scientists of this data.
So what's the greatest kind of challenges then for your research focus going forward? Is it technology? Is it policy? Is it regulatory? Is it funding? What, if you had to name maybe one of, kind of the hardest or the greatest challenge that you're, kind of faced with day to day, what is that?
We're developing the technology very actively, and there's always new technology occurring both in the industry space and academic labs. And in some way, the technology is running quicker than we actually be able to generate data with that technology, because a lot of the questions do reach funding because we develop, let's say, a single cell technology, a spatial technology. I'm developing special technologies to do single cell profiling in the laboratory, or we're doing the technologies to do profile blood, cell-free DNA and cell-free RNA. All of those are relatively expensive compared to maybe some sort of old school techniques. And we also need to analyze relatively large patient populations because, unfortunately, currently, even if we talk about cancer and even simple sequencing techniques, there's not that many individuals that have been studied, individual tumors that have been studied. For example, even large whole genome sequencing or whole exome sequencing cohorts maybe reach a few, less than 100,000. There's probably 20, 30,000 total samples that have been studied, and that's across all cancer types. So for some tumors, there's maybe a few hundred or less than 100 samples that have been studied. And each tumor in some way is actually different. So in each tumor in each patient, even though it might be a similar cancer type, it might have its own features, its own driver mutations, its own response to therapy. And there are common patterns, but we need to sample much larger populations to be able to make the sense and actually design therapies that work across many patients, not just for one patient. And really, we don't have enough of that information collected now. There's almost 18 million people living with cancer in the United States alone. And we're talking about that over 10 years, we profiled 10,000 tumors. That's really a drop, in some way, a drop in the bucket of what we could have studied. It is mostly funding constraints, and it does require a lot more work to really study. There are bigger projects that study more like heritable DNA. There's Broad Institute has a project that studies million genomes, but that's just basically genetic predisposition rather than actually studying the disease condition. So for the disease condition, even cancer, which is one of the probably most studied conditions, we still have very limited sample information. If we're talking about single cell data sets, that's even fewer. For some conditions, there might be less than a dozen samples that have been analyzed. So it's really, we're working very small data sets that are probably not enough to answer some of the more important pressing needs.
Just so I understand, the samples are there, with the people that, is it that we're not collecting the samples? Is it that people that with cancer are not releasing the approval to study those samples? Or is it really just the time and the effort and the technology usage to analyze the samples? Is it which side of that problem, or is it all sides of that problem? Is it the collection of the samples, the approval, or is it truly just the time and the money it takes to analyze the samples?
We do have large biobanks for multiple disease types, including cancer. So sample collection, at least for more common conditions, I don't think that's a bottleneck at this point. It is always hard to collect samples on a specific treatment or specific clinical trial, for example, specific cancer type times specific treatment. Yes, that's hard to collect sample, and that's why we're pushing these non-invasive methods like blood biopsy to be able to collect samples easily from pretty much any participant in any study, cancer or non-cancer conditions, because that really opens the ability for people even that are not specifically donating their blood at the hospitals. We had situations where people were willing to do like a direct-to-consumer donation that were actually interested in research and were willing to get their blood drawn and sent to a research lab. So these technologies are enabling this side of the sample collection problem. I think, yes, analyzing samples and the time that it needs, the people time, and definitely the technology that needs to be applied is relatively expensive. So so funding is really, we're kind of always short on funding in terms of actually being able to answer some of the questions. Sometimes you feel like, well, if I collect another 100 samples, I would actually answer the question, but you need funding to be able to do just that part.
Yeah. So, in respect to your research and what you do and kind of your dedication and passion, what do you hope for the future? Like, when you look out 5 or 10 years from now, what do you hope that you, your colleagues, the community has achieved?
I'm very hopeful in terms of using these new technologies and profiling disease conditions in more detail. And for the first time, maybe being able to really see the changes, the biological changes of the tissue, not through imaging studies and some sort of just outside look from the body, but actually seeing direct effects of treatment and conditions from these blood tests, and that we'll be able to unlock some of the bigger problems that exist. First of all, early detection for pretty much early disease condition. Cancer, very important. Alzheimer's disease, especially some of the therapies that are being developed, are much more effective in early stage Alzheimer's, and potentially, if those individuals are diagnosed much earlier than there's any sort of symptoms or imaging changes, we'll be able to treat the condition before it actually cause any sort of symptoms. The same for definitely for tumors. As I mentioned, I think in the next 5 to 10 years, that would be the major shift is to be able to identify tumors much earlier and being able to do screening and basically prevent the tumors from developing into something aggressive and be able to treat them. Plus, I think this non-invasive monitoring will also allow us to personalize treatment plans and have more tumor-informed treatment protocols where you actually see what's happening with the tumor or disease organ and adjust therapies as we go versus having clinical guidelines that are just kind of an umbrella for all the patients with this disease condition. So these more personalized approaches are also, I think, something we're looking forward to. So we're very optimistic. Developing drugs are very complicated and a long process, but we're hoping that if we're moving the diagnostic technologies ahead, we'll have much more biology and much more understanding of how to prevent the disease and also buy time by detecting it earlier, so more therapies and more interventions can work.
So there are a lot of people that may be listening that they don't have a researcher in their family. Maybe they haven't ever met a researcher. But they're curious about research. What do you wish those people knew about what you do and being a researcher that they may not be familiar with? What do you wish they would know about what you do?
Yeah, I think being a researcher is very exciting, actually. I think one of the most rewarding things of being a scientist is really being able to uncover new things that nobody knew before you, and really building on the knowledge of previous generations really move this kind of knowledge field ahead. And it's sort of like a detective working in a field where you actually don't know anything about the things you're studying. And with new technologies, it becomes even more exciting because now we can interrogate things that we haven't done before. And we're just opening the opportunities to really study new processes and new biology, new mechanism where we can actually study patients directly. So we're really close to studying the actual subject of the disease and understanding things that sometimes we haven't even predicted. So it's a time-consuming, hard job, obviously, and you need to train a lot. But it's very exciting and rewarding, I think, to be a scientist and to, in some way, be responsible for your own discoveries and understanding how things work.
Yeah, that's great. And you're in this hybrid lab that you discussed and kind of described. I would imagine that collaboration is very important. A collaboration across multiple scientists, researchers. How important is collaboration?
Collaboration is very important, and I could say that almost all of our studies, all of our projects actually involve active collaboration. Especially since we're working in a field of clinical science where we're actually studying disease and directly in patients. We almost always have a clinical collaborator, either a doctor, oncologist, or a clinician working, a pathologist working in a specific field. They're domain experts. They know the disease. They obviously interact with patients. They're able to collect the samples, and also they often are able to utilize our research that we uncover directly in their future clinical practice in a way that it influences their thinking, their understanding of the disease. And we often see, even with cancer treatments, for example, we see people changing their paradigm of the approach to treatment just by knowing the genetics and knowing what's happening behind the scenes, basically, with a tumor. That's true for any other condition too. So we're always collaborating with clinicians. We need to collaborate often with disease experts. My lab studies many diseases, even I would say each cancer type is a different disease because it has a very often distinct treatments, distinct biology, distinct survival, distinct approaches to treatment, even the distinct processes that cause cancer. So we often have to collaborate with a disease expert. There's labs that work only on breast cancer or only on brain tumors or only on Alzheimer's disease. And we almost always collaborate with one of those labs to be able to validate our findings, to put them in context. We often try to design some interventions if we can. So let's say we find some gene or something that is broken or modified in a specific disease, we would want to try to see if we can replicate that in the cell line or mouse model and see if there's possible to address those issues. And then eventually we try to somehow move it into clinical trials and test therapies or test approaches to therapies. My lab often works on trials that are testing new drugs in different conditions and trying to understand how these drugs work, new cancer therapies or other therapies, to understand what's happening with the tumor as it's treated, why is it becoming resistant, how we can overcome this, what's happened with the immune system and interactions with all these conditions, et cetera, et cetera. So collaboration is really driving a lot of the science we're doing.
Yeah, that's great. Well, we're wrapping up here, and I think I could spend a lot more time just kind of picking your brain and trying to understand what's going on out there. But as we wrap up here, in kind of that vein of collaboration, how would people get a hold of you if they're interested in finding out more about what you do, what your lab does, or if they're wanting to collaborate? How would they get a hold of you?
My lab runs a Twitter account that we try to post kind of new research and new discoveries from our lab and events. I often been reached out through LinkedIn, and I also try to, update about events and papers and projects through LinkedIn, and that's probably the best way to reach out. Obviously, there's a website, and you can reach out through the website. Very happy to talk to anybody. Who are often contacted by various groups throughout the world about our methods and technology and collaborations. So we're always excited to talk to anybody that is interested in our research.
Well, thank you very much. And thank you so much for the time today. I know, you've got a lot on your plate, but sharing your perspective, sharing your kind of vision for the future is very important. So thank you very much.
Thank you for having me. That was exciting.
Thank you for joining Research Renaissance. For more information and to stay updated, subscribe for more episodes and visit our website at tofflertrust.org. Feel free to call us if you have any thoughts, suggestions, or just want to find out more about the Karen Toffler Charitable Trust. Until we meet again, onward and upward.